So I'm going to talk with you today mostly about two studies that we've done on cover crop mixtures and soil health, and then a little bit about a larger scale project that's ongoing at the end. I might pause about halfway in. There's sort of a logical stopping point if you have questions, or I'd love to hear and learn from all of you about what these data make you think about in terms of nutrient management. So I'll pause and see if there's anything about halfway. But if you're confused at any point, feel free to raise your hand and I'll answer questions, and we can also have discussion at the end. So how many people in the room use cover crops? I'm just curious. Okay, so we've got a lot of experience here. What about mixtures? And by mixtures, I just mean two or more. So a handful of mixtures, great. And so you're probably aware, and this conference has told us a lot about this, that cover crops are multifunctional, right? Depending on the cover crop type and the species that you select, you can get a lot of different benefits from erosion control, nutrient scavenging, nutrient supply in the case of legume cover crops, pest and disease control, and many more functions. And there's actually a pretty large and growing body of research on cover crops in addition to growing use on farms. And what that research has shown us is that the benefits of cover crops tend to be greater if you focus on what's called functional diversity. So that means pairing species that have traits that complement one another, right? So legumes and grasses and brassicas, summer annuals and winter annuals, or annuals and biennials even. So thinking about how you can mix and match covers that have traits that will complement each other, and that means that you're more likely to increase overall capture of resources, things like light, water, and nutrients that lead to overall greater cover crop growth and potentially increases in crop yield and soil health. And in my research group, we're focusing in, and this is what I'll talk about a lot today, on two particular functional types of cover crops, so the role of legumes in supplying nitrogen to fields, and also a focus on overwintering cover crops, so cover crops in the winter niche, because we know from a large number of studies, those tend to have the greatest benefits for things like nutrient retention, organic matter accrual, and other functions. So that's the focus today, thinking about how to diversify crop rotations with mixtures. I'll mostly think about complementarity between legumes and grasses, and again in the winter niche. So you probably all know grasses are great at nutrient scavenging. They can recycle, especially soil nitrogen and other nutrients. Legume cover crops can also scavenge nutrients, but they have a different function that's supplying a new source of nitrogen from the atmosphere through biological nitrogen fixation. And then again, overwintering cover crops are a focus here because they build organic matter and reduce nutrient losses. And before I tell you about the research in the two studies, I'll just give a very brief refresher on what I'm talking about when I focus in on legume nitrogen fixation. So legumes are really cool. They have special bacteria, rhizobia, that live in these nodules that you can see if you squint. They're really tiny on this screen on the roots of those plants. And the rhizobia convert or fix nitrogen gas that's up in the atmosphere into forms that plants can use, specifically ammonia. And they do that in exchange for carbon. So they give carbon to the bacteria, and the bacteria gives the legume plant nitrogen. So what's great about this is it's essentially a solar-powered source of nitrogen on your farm. And the way you get that nitrogen is when the cover crop is terminated and that legume material decomposes and breaks down, the nitrogen is released. And some of it ends up in your soil organic matter pools, and some of it ends up being available to the crop that you grow next. So with both of these studies, the goal is the picture that you see here. We know from a lot of work that cover crops improve soil health. And here we wanted to ask the opposite question. How does soil health affect cover crops? Because across farms, we have very different management systems, different management histories, and different baseline levels of soil fertility and soil health. And so you might expect that that will affect how a cover crop establishes and how it grows. And the growth and establishment of the cover crops will determine the functions or the on-farm benefits that you get. And in this work, we were focusing in on nutrient-related benefits of nitrogen retention and nitrogen supply from the legume, among a couple of other things you see there. And then the hope is over time, if we learn more about this, we can incorporate feedbacks to management that help us tailor how we manage cover crops on farms that have different conditions. So hopefully that pathway makes sense. So the first study I'll tell you about was a field station trial. This was at the Kellogg Biological Station in Hickory Corners, Michigan. It has a long-term cropping experiment called the MCSE, the Maine Cropping Systems Experiment. It's been in place for 30 years now. There's a lot of treatments, but we focus this on the 4 annual cropping systems. These are in a corn, soybean, winter wheat rotation. And there's a conventional system, a no-till system. The reduced input system has 1/3 of the pesticide and fertilizer inputs of the conventional system. It also has rye and clover cover crops at different points in rotation. And the organic system is certified organic. And what's interesting about that one compared to some other long-term studies is the only external input in that system for the past 30 years is a red clover cover crop that follows winter wheat. And that system also has rye after soybean. So we took a baseline soil health sample, fully characterized what soil health was like in those 4 long-term management systems. We then planted into that our cover crop treatments, so a Sol rye, crimson clover, the rye clover mix, and then a weedy fallow control with no cover crop. And maybe you can see the seeding rates there, 90 pounds per acre for rye, 15 for clover, and then 45 and 8 for the mix. So in the conventional and no-till system, that simulates a transition, the first time ever putting cover crops into those systems. And then for the other 2, that mixture is a diversification of the cover crop. They've never had a mixture before. We planted those at 7.5-inch row spacing after the winter wheat harvest, and they were terminated between May 13th and June 1, depending on the management system. That late date was the organic system because it has a later planted variety of corn. So these numbers you obviously can't see from where you're sitting. I'll just tell you the point that I want to make, which is just I want you to believe that, in fact, these systems did have different starting levels of soil fertility when we went into this experiment. So it says conventional, no-till, reduced input, and organic. And first I'll just point out at the bottom here, that's texture, sand, silt, and clay. No difference. It makes sense. It's one small site. It has the same soil type. But these are different biological and chemical indicators of soil health, where bold font means it's higher. And so overall, there were higher levels of soil fertility and soil health in the reduced input and organic system after these 30 years of the trial and use of cover crops in those 2 systems, with the exception of phosphorus, which is quite low in that organic system, again, because the only input is the red clover cover crop. And what was interesting was that those differences in soil health across the 4 systems, those panels are conventional, no-till, reduced input, and organic. And this is showing you above ground biomass of our cover crop treatments in tons per acre. And the main thing I want to show you here is that soil health affected how those treatments grew and the composition, especially of the mixture. First of all, rye biomass was quite a bit lower than we expected. Probably those of you who plant rye often would say, well, not a great idea to plant it so early maybe, especially with the summer conditions that we had. But generally, clover did much better in the 2 systems that had lower soil health. So there was much more clover biomass in the conventional and no-till system. And that follows what we know from research, which is that clover thrives in lower fertility conditions. And then interestingly, the mixture was much more even in the, is there a question here? Yes.
Reduced input, are you talking about like a vertical till?
It has full, what is the tillage there? It has conservation tillage, and the inputs are roughly 1/3. It's mostly the pesticide and fertilizer inputs that are different, so they're 1/3 of what's in the conventional system. And so what was interesting about this, the cover crops didn't perform that great in that system, except that the mix was much more even, which was interesting to me. And that system does have higher biological soil health. And in the organic system, we had the greatest biomass and the greatest evenness. And so the main thing I want to point out here, just for us to think about as I then move into the on-farm results, is that even at one site, the mixture differed pretty widely across the cropping system, so that's interesting. And that higher soil health meant higher cover crop biomass overall, and a more even mix of the clover and rye. And just to show you that, these are 2 biological indicators of soil health, particulate organic matter and mineralizable nitrogen. And that was positively related to evenness of the rye and clover. So a number 1 at the top there is like a perfect 50-50 mix of clover and rye. So that evenness of the two species increased with biological soil health. And evenness was related to a couple of benefits that we measured. Greater nitrogen uptake by the following corn crop, a higher C to N ratio of the cover crop biomass. We did a bunch of nitrogen measurements in this study, and it seemed to slow the timing of decomposition enough to better match that end release with the corn nitrogen uptake. Evenness was related to more of the clover nitrogen coming from the atmosphere, so making the clover work harder, getting more out of that legume. And a greater % recovery of a bromide tracer that we added, and that's just an indicator of potential for nitrate loss or retention. And you can't really see that well here, so those are just those a few of those relationships in graphs. And I'll skip to the takeaway from this, which was, first of all, the mixture benefits depended on the management system and the properties, soil properties going into the study. And so from this particular site, if we were to turn this into a recommendation, I'm curious to hear your reaction to this, it seems like it makes sense to actually start with individual functional types of cover crops in the early phases of transition, so a sole legume or a sole grass, at key points in the rotation, and then move to mixtures as soil fertility starts to build, to be able to really maximize the benefits of those mixtures, given that the seed costs are slightly higher and they're more labor to plant. And then I want to share with you the nitrogen results, because I want to focus on nitrogen a bit today. So across all the management systems, the sole rye scavenged 5 to 24 pounds of N per acre in the above ground biomass. Again, we had sort of low rye biomass here. Clover accumulated 18 to 79 pounds of nitrogen per acre in the sole stand. And then the mixture had 17 to 73 pounds of nitrogen per acre. So a similar amount of total above ground N to the sole clover. But then what we're able to do is figure out, well, how much of that clover nitrogen represents fertilizer, like a fertilizer input from the atmosphere. And we use what are called stable isotope methods to do that. So we look at the ratio of the heavier to lighter isotope of nitrogen to figure out the source of the nitrogen in the plant. And so across the systems, that number was 10 to 40 pounds of nitrogen per acre. So that represents the new nitrogen input to the field. And the only other thing I'll point out on this is that generally that number tends to be higher with a sole legume stand, simply because you have more biomass compared to when you're growing a legume in a mixture. Any questions on that? I think I'll just move right on to the on-farm trial, because the results were, we asked a very similar question and the results are a little bit more interesting because this is on actual working farms. So here we had a similar focus on how do different soil conditions across farms affect cover crop performance. This time it was on 14 organic farms, not all certified organic, some were in transition. They were in 2 regions of Michigan, the southwestern region and the Thumb. And here, same idea, except we did a 4-way mix instead of just 2 species, with this same perspective of trying to maximize functional complementarity, having species with traits that contrast and complement one another. So we had a legume, a grass, a brassica, and we had 2 grasses. One was a summer and one was a winter annual. So rye, crimson clover, oat, and dwarf Essex rapeseed. Maybe if you squint, you can see the seeding rates there, 28, 25, and 2. We planted the sole cereal rye and the sole crimson clover as our comparisons, and also a no cover crop control. Oh no, not in this one we didn't have a no cover crop. But what we did have was a farmer choice mix. So each farmer also selected their own mix of their choosing, and I won't share those results today, but it was a fun part of the study. So each of those treatments that you see there were replicated 4 times along the edge of a field in plots that were roughly 20 by 120 feet. And we did this over 2 years in 2 different fields. And so the fields were coming out of a small grain, usually winter wheat, and then we went into these treatments. And we did a complete soil health assessment before planting the trial, so biological, chemical, and physical indicators of soil health. And these are the biomass results. So this is looking at year 1 and year 2 of this trial, the average across all of the 14 farms, above ground biomass in pounds per acre this time. So the blue bar is rye, and you can see rye had about low biomass again. It was the same across both years, 1,700 pounds per acre on average in year 1 and year 2. The gray part of the bar are weeds. These are organic farms. They have significant weeds. The green bars are the crimson clover. Sol clover did pretty great in year 1. We had 3,700 pounds, I think. Oh no, 3,000 pounds per acre of sol clover in year 1, and then only 1,800 in year 2. And then the four-way mixture performed the best of all of the treatments in terms of overall biomass production. Especially in that first year, we had 4,500 pounds per acre of mixture biomass, and in the second year, 3,700 pounds per acre. So a slight decline, but again, pretty stable over the 2 years. And in the second year, you can see by looking at the colors there that there was a more even mix of the 4 species in the second year, which again is important for really, you have to have representation of each of those species to get the benefits that they can provide to the farm. These farms had, they had a wide range of soil health, like a range of starting soil fertility levels, but because they were mostly organic, I would say almost all of them had a history of using cover crops. So thinking back to what I was saying about the first trial, these folks there is a history of cover crop use on the farms, relatively good starting levels of soil fertility, which might also contribute to the really strong mixture performance here. So across both of the regions and years, what did we find for the nutrient results? So then this is the nitrogen that was recycled in the above ground cover crop and weed biomass. In rye, there was an average of 34 pounds of nitrogen recycled. In clover, an average of 72. And in the four-way mix, an average of 62. And you can see the ranges across the farms there, so some were quite a bit higher and some were lower. So, you know, up as high as 116 for the four-way mixture. But this is also an underestimate because that doesn't count roots, right? Roots are doing quite a bit for us as well. And thankfully, I have a really ambitious PhD student who actually wanted to sample roots as part of her dissertation. Labor intensive, so she did this in just one of the regions, so half of the farms. And we're still processing the data from year 2, so I'm going to show you what we have for year 1. So keep in mind this was the better performing year. It's hard to see what the different shading is, but it doesn't matter very much. Those are just different size classes of roots going from fine to coarse roots. So if you look at the bar for mixture on the left, that light part of the bar at the top is the coarse taproot of the dwarf Essex rapeseed. So having that functional complementarity leads to overall greater root biomass in the mixture, almost twice as much root biomass as in the sol, clover, and rye, which had comparable levels of root biomass. And then the panel on the right is the amount of root nitrogen. So even though clover had the same biomass as rye, it had more root nitrogen, which makes sense because it's a legume. So on average, I think there were 23 pounds of N in the rye roots, 31 pounds of nitrogen in the clover roots, and 43 pounds in the mixture roots. So more overall nitrogen in the mixture, but you know, again, a fairly substantial amount. And so if we add that together, roots and shoots, that changes things a little bit. The sol rye is 58 pounds of nitrogen per acre, and the clover and mix are both around 115 to 116. So fairly substantial amounts of nitrogen captured by these cover crops on average. But again, the clover is unique. So for most of these species, that represents, you know, taking up nitrogen that's already in the soil and recycling it. But for clover, some of the nitrogen is from the soil, and some of it is from the atmosphere, which is a different function. That's essentially a fertilizer input. And so again, we quantified that for these sites. We used isotope methods to figure out how much of that clover nitrogen came from the fixation activity of the bacteria, came from the atmosphere. And so on average, this is just the above ground, it was 51 pounds per acre for the soil clover and 23 pounds for the mixture. So there's that whole benefit of the nutrient recycling, and then this is the portion of that that's a fertilizer input to the field. The biomass was lower in the second year, and so that input was lower in the second year. So for mixture, it was about half of what it was in year one, because the biomass was about half of what it was in year one. So I think that says 25. This is testing my vision. And then the mixture was 16. And because the mixture had more stable biomass over the 2 years, that amount is a bit more stable. And so for me, one thing that's interesting here to think about are just, the trade-offs with management. Depending on management goals, it might make sense to have a sole legume stand if there's a big need for a nitrogen input or wanting to offset input costs. Whereas the mixture provided a more stable amount of nitrogen over the 2 years, but also other functions. I don't know if you remember from the biomass graph, but there was less weed biomass in the mixture and more nutrient scavenging compared to the sole clover. This doesn't include the below ground portion. That's harder to do with this approach. So I'll just give us maybe a conservative estimate. You could probably add, based on our below ground numbers, 23 pounds of N to that sole clover component, and maybe 7 to 8 pounds to the mixture, but that's a little less certain. So when we add that in, again, fairly substantial. So before moving on to the next piece, I was just going to pause there for a moment because I'm curious just if there are questions or if that's clear, or if anyone wants to share their own experience or react to that, those nitrogen numbers.
My question would be, with the biomass above ground, how much of that do we really capture?
Yep.
Feasibly?
Right. I was going to get to that next, so good question. So the question was, all of this is tied up in the biomass, right? So how much of that do we get in the next crop? Which is a hard question to answer, but I'm going to take a stab. Any other comments before we move on? Mm-hmm. Yeah, so what's interesting, if we think about shoot versus root dynamics, I would say the roots, because they're in close contact with soil microbes and soil particles and aggregates, their carbon and nitrogen is more likely to be stable and to stick around in your soil and enter different organic matter pools. It won't be released as quickly. Yeah. The shoot material is going to decompose more quickly, but also that means some of it is more subject to being lost. Any other reactions, or we can have more discussion at the end. I can plow ahead. Just curious if that was clear, if there are any questions. Okay, so coming back to this question, which is an important one, I'll first just show you this relationship, which is common, which is that crimson clover biomass across all of the plots on the bottom, and the amount of nitrogen fixed, that nitrogen from the atmosphere on the y-axis. So those things are very strongly related. So if you need a nitrogen fertilizer input, well, the more legume biomass, the better. This also means, we used expensive methods to actually quantify that very precisely, but you can estimate it pretty well just by measuring your legume biomass. And I have a handout with some ideas on how to do that on your own farm. If anyone's interested, we've given that out to farmer participants, so we could share that with you. But it involves, cutting biomass from a known area, let's say with yardsticks, and then drying it, and ideally having it analyzed for nitrogen. So coming back to this great question, can legume cover crops supply all or part of your nitrogen needs? We need to know how much of that residue nitrogen is available in that first year or growing season. And I'm going to be disappointing and say more work is needed. We don't fully know the answer to this, partly because it hasn't been done across a lot of different, so I'm talking about a study across a lot of different farming conditions, and this is what we need. But work like this, which is more expensive and labor intensive, hasn't really been done across a lot of sites. But we do know some things. I would say, again, isotope methods are probably the most precise way to know this. So these are studies that have labeled an actual legume with the heavier isotope of nitrogen, and then they track where it ends up, like how much of it is in the following corn crop. For a number of reasons, it tends to be a very slight underestimate, but on average, 20 to 40% of legume nitrogen is recovered by the following crop in the first year. Another approach people use are litter bags or other ways to measure loss of the mass over time. A little less accurate, perhaps a slight overestimate, but those on average are about 40 to 50% of the residue nitrogen decomposes in a growing season. So somewhere between 20 and 50%. I know that's not super satisfying, and that's why I say more work is needed. That variation depends a lot on your soil conditions. More will be released with higher soil health because you'll have more biological activity, and decomposition is just ramped up. And so that's the main point I want to make here, is that regardless of what that number is in, say, years 1 to 2 of using cover crops, with continued use, let's say you get to year 5, 6, 10 of cover crops, legumes are building your microbial biomass. They're building a lot of different soil organic matter pools that just increase overall mineralization. So I would feel much more confident if you're in year 5 to 10 of a lot of cover crops in rotation, saying, well, you could back way off of inputs, than in the first year or two, where it's a lot less certain, and you're probably getting 20 to 40% of what's in. And it, again, depends on how much legume biomass that you have. So this is an active area of work, and I feel like for folks to feel confident managing biological sources of nutrients, we need to know more about this, but it's hard to measure. And so thinking about this type of question, we had a range of soil properties across all of these farms, which allowed us to start to dig into, well, what explains some of these differences in performance? And when we looked at the clover biomass in both mixture and monoculture, we found that overall clover biomass increased with higher plant available phosphorus. And this, I think, is fairly well known. Clover can supply its own nitrogen, so it tends to be limited by phosphorus. And legume biomass decreased with soil nitrogen availability. So when we had higher levels of soil inorganic nitrogen, nitrate and ammonium, when we had higher biological measures of nitrogen cycling and nitrogen availability, we had lower clover biomass. And I think that's a cool result because it actually shows the built-in efficiencies of legume nitrogen sources. They tend to lose less nitrogen than something like synthetic fertilizer or compost, in part because legumes self-regulate. If there's a lot of nitrogen in soil, they don't want to pay that carbon to their bacteria, and so they'll play more of a scavenging role. But if soil nitrogen is low, they'll invest more in fixing new nitrogen from the atmosphere. Maybe from a management perspective, this also means if you have a lot of nitrogen in your soil, you might not need to pay for legume seed unless you want them for another reason. Maybe they're flowering or forage potential. And then in contrast, when we looked at rye across these 14 farms and all of the plots on the farms, rye biomass increased with soil fertility and soil health. And this, again, might be expected. So especially these metrics were good predictors of rye, so CO2 burst test, so mineralizable carbon, potentially mineralizable nitrogen, plant available phosphorus and potassium. So rye biomass increased with all of those things. And again, there's sort of a positive feedback loop we could think about here, where as your fertility builds, you get more biomass, greater cover crop performance, greater nutrient scavenging, and more recycling. So again, to me, this highlights that there is more uncertainty in those early years of transition, and we need to provide more support to folks who are just starting to cover crop, and that over time you should start to see these benefits and these positive feedbacks. And so I'll just end that part by saying we need more work that helps us give better nutrient management recommendations based on all of this biological complexity. So this figure here is from a different paper in a different study, and what it's showing is the CO2 burst test on the bottom and how the corn economic optimum nitrogen rate decreases as CO2 burst increases. So with higher soil health, the nitrogen inputs can be lower. And we have a similar relationship in this study. But there's a lot of variation on that graph, right? That's not very certain if you're trying to manage your nutrients. And so we need to figure out what are the soil health tests that are a better predictor of nitrogen supply, and also measure those changes regularly over time in fields. And this is not an easy task. Understanding what we call bioavailable nitrogen is actually really complex because of all of these interactions between plant roots and soil microbes and soil minerals and soil organic matter. So it's a work in progress, but I would say this, this certainly represents progress. And in addition to that, I would add, to do this well, we need to know your legume biomass. So keeping track of, how much biomass is actually there to understand the actual nitrogen input. And then related to that, that allows us to construct more accurate nitrogen budgets. So you fully know the total N input from fertilizer, legumes, or manure, and then can subtract the amount of nitrogen that's removed in harvested crops over time. So over time, those things should approximately be in balance for optimal nutrient management. Pause again in case questions or comments, related experiences. I think so. Yeah, I think this is an attainable goal. This isn't terrible. Like I'm saying, there's uncertainty here and this is hard to do. But not a lot of research has been dedicated in this area. We've done a great job of fine-tuning fertilizer management. But legumes have not gotten the same love. And so I think, it's gonna take a little more time. Oh. Potentially, yeah. So I'm cautious about suggesting that we should add fertilizer to cover crops because I feel like the main point is to try to scavenge and reduce those losses that are problematic. But in the next piece of work that I'm about to show you, we did find higher cover crop biomass on average, slightly higher for folks who fertilize their cover crops. And so, yeah, I think in the early years, or maybe it's supplementing with a little compost or something, especially if you're trying to get a legume to grow. Maybe it needs some phosphorus. So yeah, that's certainly a possibility. But again, that's why I really think that nutrient balance perspective is important because you don't want to do that 10 years in. That's just, a waste of that input cost. Yeah. Any other questions or comments before we move on? Okay, so the last piece is I'm going to shift gears just a little bit and talk at larger scales about cover crops and cover crop biomass because I've been trying to drive home this point that, well, if you want to get the functions of a cover crop, it needs to put on some biomass, right? And these two pictures on the left here were from that on-farm study. So they were taken in the same county. We used the same planting method, the same planting date. And you can see really different levels of cereal rye in the spring. And this might be a common experience if you use cover crops on your farm. There's variable outcomes. And so my PhD student, who's pictured here, Etienne Sutton, she's leading some work that we're doing from a citizen science approach. So trying to figure out across the Great Lakes region, what are some of the main factors that are driving this variable performance in cover crops in different farms. And so you can see a lot of different factors listed here that we know matter, things like climate, soil properties, topography, nutrient management, planting date, planting strategy. And so by partnering with farmers are actually helping in the data collection effort here. So that allows us to include a large number of studies in this project. The goal is to identify some of the main environmental and management factors that explain variation in cover crop growth, in cover crop biomass. And by better understanding that, we hope that can help improve site-specific recommendations to figure out how we can maximize benefits of cover crops in different settings. So this is really building on what I was just talking about, thinking about what do we need to know to really tailor management to different sites, but doing this at a much larger scale, thinking about the whole region. Here's my commercial. This is the sales pitch. More farms will mean better recommendations. So I know some of you are actually participating in this, but if you're not already, we'd love to have you sign up. There's still time to sign up this spring, and I'm going to explain what that entails now briefly. If you have a field with cover crops that are overwintering, then you're eligible to participate. So the approach to doing this, the way that we're collecting data, is the farmers who sign up are giving us 2 pieces of information, all anonymous. The first is a short survey about management practices specifically on the cover cropped field. So main things that we think will matter, like planting method and planting time, all of that stuff, the field history. And then the second thing is to perform a simple field assessment. It takes about 15 to 30 minutes, depending on how big the field is. And we're using that to estimate cover crop biomass. And that picture on the bottom is showing you what that entails. So basically, you go out in the field and take a picture, sort of with your phone facing this way, of the cover crop canopy at 3 different locations in the field. And then we run that through a software program called Foliage that calculates the percent cover. So in that example picture, there's 68% cover of cereal rye. And then you take height measurements of the cover crop in those 3 locations. And so when we multiply percent cover by average height, that's a good predictor of cover crop biomass, or a good estimate of cover crop biomass that allows us to have a really large data set, because this is much less labor intensive than going out and clipping all of the plants with scissors, which is what we typically do. And so in last year, these are the preliminary data from year 1. This is going to keep going so that we can get a really nice database. So last year we had 129 fields that were in this work in Michigan, Ohio, Indiana, Illinois, Wisconsin, and Minnesota. 62 of those fields were in a single species, mostly rye, and 65 fields were in mixtures. Most of those were in row crop rotations. And then we want to figure out, okay, well, how accurate is that estimation protocol that folks are doing, the pictures and the height measurements? So Etienne went to a subset of more than 60, I think it was almost 70 of the fields with some research assistants, and did the tedious thing of clipping the plants with scissors and weighing them in 3 different spots in the field. And so we use that method to validate the height and cover measurements. I know you can't read that text, but this is a strong relationship, explains more than 80% of the variation. And so we can use that equation there to estimate actual biomass for farms where we don't clip with scissors. And so that's the way that we're estimating biomass across this large area. And then again, for my sales pitch, there's sort of a low number of fields from Ohio last year, 12. And we have to do the validation again. So my plea is, don't make Etienne drive to Wisconsin again. Let's have more in Ohio, which is our friendly neighbor much closer by. These are the initial results from that first year. On the left is the sole planted cover crops, mostly rye. And the red bars on the right are the mixtures. So overall, one interesting thing from year 1 was that the mixtures were slightly less variable. So going back to what I was saying in the other study, the mixture biomass was a bit more stable, less variation across all of these fields. And this bottom here is the average biomass. It was slightly higher for the mixtures, 900 or about 1,000 compared to 900. But that's the second point that I want to make. Overall, this biomass is pretty low, right? You can see there's a lot of variation, which is what we were wanting to get, so we can start to figure out what explains that. But a lot of the fields were on the low end. And I think, if we want to have the benefits of cover crops, they need to actually put on some biomass, and this goes back to that there's a lot of pressure to terminate early and plant the spring crop. And so that can really constrain the benefits of cover crops. But again, we want to figure out, okay, well, what does drive this variation, and why are some of the fields really on the high end? And for rye, some of the best predictors were things you might expect, like more rye with more growing degree days, more precipitation, with greater seeding rates. There was less rye with later and later planting dates. We also were able to use that farm and management survey to look at some of the management practices. So here's just one example, the seeding method. Rye is on the left, and mixture is on the right. And on the left, that's rye biomass per growing degree days, so normalized by growing degree days. And you can see that rye did much better when drilled compared to broadcast or aerial seeding, much higher biomass. But for mixtures, there was actually no benefit of drilling. So the same biomass for mixtures, for broadcast and drilling. That's a good question. I don't know offhand, but we can look at that in the survey. That's a good question. So it was, when broadcast, was it actually incorporated a little bit or just left on the surface? I'll make sure that's in there. With the validation samples, so where Etienne went and clipped the biomass, we're also analyzing those for their nutrient content, so for carbon and percent nitrogen. And so we can give that back to participants, and it also allows us to understand the nutrient scavenging and a little bit more about the nutrient dynamics. Here I'm just showing the average carbon to nitrogen ratio for a subset of the samples that she clipped. So for rye, wheat, crimson clover, hairy vetch, rapeseed, turnip, and then weeds. And the main thing I want to point out here is just that these C to N ratios are really low. They're all well below 25, right? Like about 11 to 16. And so I think that matches with that other graph I showed where we had a lot of low biomass fields. So the downside is, if the cover crop biomass is so low, they're probably not doing a great job of nutrient scavenging. And then, in turn, we have this lowish C to N ratio, which, maybe on the one hand, that's a good thing. You're certainly not tying up any nutrients in this situation. But it could, again, lead to the problem where the little bit of nutrients that are scavenged are released really quickly and not timed well with the growth of the next crop. So these are the types of things we need to dig into more. And having a lot more data and doing this over years, I think will be really interesting to start to figure out, what are the best approaches for cover crop management in different contexts. So this is, again, the commercial. We're still recruiting for this year, and the more fields, the better these data will be, and I think it could lead to some really helpful information. So if you're really tech savvy and know about QR codes, you can take a picture here. And that will lead you to a survey to just sign up. Or if you prefer, you can just email Etienne. It's at the bottom there, herrick.umich.edu. Or if you miss all of that, just come see me afterwards and I can help out. So again, really hoping we get more folks from all states in the Great Lakes. And then I might be ending a little early. I'm not sure where we are on time, but I didn't want to bore you with a really long monologue. So I'll give you the overall takeaways, and then maybe we can have some discussion if there's time. So 4 main takeaways that I wanted to get across today. The first is the importance of overwintering cover crops, or the role that they play in scavenging and recycling soil nitrogen. And also, when there are legumes planted, either as sole legumes or in a mix, they also provide a new source of nitrogen to fields, which is important to understand and account for in our nutrient management. And these benefits of overwintering cover crops depend strongly on biomass production and successful establishment of the cover crop. And we're in multiple projects trying to understand more about what explains that across farms. The second main point is the legume nitrogen supply especially depends on biomass production. So we have more total nitrogen typically when you plant just a sole legume cover crop. But what's interesting is legumes work harder. They fix more of their nitrogen when they're planted in mixtures. And so that goes back to what I said in the very beginning, that there are ways, when managing diversity, crop diversity on farms, you can harness these complementary traits or interactions of species to really maximize their benefits. So this is sort of a cool phenomenon, and again, depending on your management goals, you might want one or the other, a lot of nitrogen, or maybe a smaller amount of nitrogen, but these other interesting functions of a mixture. And overall, we found that the mixtures were a bit more stable in their performance over time, and that makes sense because in different conditions in different years, well, one species is more likely to succeed in a mix than if you just have only one thing planted. We've found that measures of soil health, which are slowly becoming more common and more available, explained differences in cover crop biomass across farms in the field station experiment, or systems in the field station study and across farms in the on-farm study. And that soil health also explained mixture composition, which is pretty interesting. Which functional group was more dominant, the legume, the grass, or the brassica? And then finally, all of that, I think, points to what I was saying earlier, that there's a need to refine and develop better nutrient management recommendations that account for all of these dynamics, that account for the amount of nitrogen that's recycled by cover crops and cover crop mixtures, and also think about the new nitrogen input that's coming from the atmosphere and how that differs in sites that have different soil properties, different management histories, and different management systems. So I'm going to stop there and just thank a subset of people in my research group who worked the hardest on the projects that I talked about today, and then especially the 14 farms that participated in that 2-year study. That was a really big commitment on their part, and they allowed us to collect some really nice data. And also the Kellogg Biological Station, some folks at Michigan State, Sig Snap, Vicki Marone, and Robin Casey, and funding from the USDA. And I'll maybe go back here to pause. So we're at just a little bit after 10 o'clock. I think we have about 5 or 10 minutes. Is that right, Sarah? So plenty of time for discussion. I also would love to just hear reactions or comments too from folks who actually manage cover crops.
Is there a place for like late planted soybeans as a cover crop?
I think so, yeah. And so by that you mean not harvesting for grain? Yeah, absolutely. I think soybeans do make a nice summer annual option. Has anyone else tried a cowpea or other summer? I didn't actually harp on that point, but when I've talked with different farmers, sometimes we use cover crop, the word, in different ways. And there are some folks who, let's say, would harvest winter wheat but still call it a cover crop. And I don't necessarily, in the way that I was talking about it here, because that does have big implications for how much of that biomass nitrogen is returned to soil. So it's still providing an important role of winter cover and reducing erosion and scavenging some nutrients. But when you harvest, a lot of the nutrients are removed with the harvested portion of the plant. So all of this accounting would be different. And we actually have a new study that we're starting focused on cover crop grazing, which I know is of interest to folks, just to think about, when you graze, what are those dynamics? How much of the nutrients stay in place if manure is distributed in the field versus what is the trade-off with some of the nutrients going away?
I've got a question. What do you think the impact of, say, herbicide carryover is on, say, your cover crop establishment? And along with that, maybe another factor would be, say, low moisture in the fall. So can you comment on those two factors?
Yeah, I think they both matter. So certainly herbicide carryover can be a factor. It's not something I study in my work. I wonder, there might be a way we can pull that out of this citizen science work, and thank you for raising that, because maybe we'll dig into that more. And then absolutely fall moisture for sure, and I think that really impacted our results in both of for the rye in both of those studies, because we planted them at a very dry moment. And in the year when we had, I can't remember if I said this, but in the year where we had lower biomass, the second year of the study, we actually had a really nice fall. It was like a wetter fall and warmer, but then the spring ended up being too wet and too cold, and so that's what happened in that second year.
I think your statistics showing that when you do cover crops for the first time, having a single species is justified. We always said that, so it was good that you put numbers to that. My other point would be, ever thought about using biosolids and manure to see what bang for buck you can get for nutrient?
Yeah, so I think we'll look at that a bit in this cover crop grazing work. It hasn't been a focus of what I've done to date, because I've really been interested in the legume question. But I think there are important interactions with manure, especially that phosphorus input, really boosting legume biomass. And so yes, that's a great point.
In the past, I've used cereal rye and then on ground going to corn, a four-way mix. Excuse me. This year I'm looking at maybe trying some, as many as 12 species following some wheat. Any recommendations on that?
Yeah, I think so. From a research perspective, there's not super strong evidence that more species are necessarily better yet, but it's partly because of these variable outcomes and that they don't all necessarily establish. But what I would recommend if you are planning like an 8 or 12 or 14-way mix is that you still go for that target of having the different functional groups of legume, grass, brassica, and summer and winter annual, and maybe trying for like 2 of each or 3 of one category, and then at least hopefully 1 of each category will do well. And so it is a little bit of an insurance hypothesis, right? When you're doing that, well, at least 1 of each category will hopefully do well, and you'll still end up with greater overall resource capture and biomass than if you just had a sole cover crop. But there isn't like strong evidence that say 12 is so much better than 8, or and that depends. Sorry, shoot.
Our big concern in this area is getting a cover crop out after corn. Have you done any studies with interseeding, say at V7 or 8, when corn's, and will a 4-way mix grow?
Yes and no. I don't have a lot of experience with that yet. We just started a new trial at the Kellogg Biological Station. It's part of the USDA's LTAR network now. And so that's a main component of a new rotation that will be studied there over many years. So more information to come, but there's a regular point in that rotation with interseeding a mix into corn. We have some of that represented in the citizen science project, so we can look at that a little more. I mean, that initial year showed that when it was aerial seeded, it had much lower biomass than for folks who were able to drill it in. So I feel like we have some great farmers who have informed that work at KBS, and I think they could say a lot more than I can about what exactly has worked. So I feel like in talking with farmers, there are folks who really get that down. And some of them have told us that they actually interseed earlier than we would think would work as researchers. So I could maybe make some farmer connections for you, but I don't have a wonderful answer to that myself.
So one question on the legumes on your study. Do they use inoculants or non-inoculated?
Yep. They're
Yeah.
inoculated, yeah.
So you have to be inoculated.
And I definitely recommend inoculation. Especially, I mean, we had this question earlier about the herbicide carryover, but especially if you don't have a history of growing legumes in a field, that's really important for making sure that they're actively fixing. If you've been growing legumes for decades and decades, it's a little bit less important because there will be active populations of rhizobia in the soil.
When you would put a mixture of cover crops out, I think it's always good to put a few tillage radishes out there because of the compaction. Compaction is a triple thing, and it's a good tool other than metal to loosen that up.
Yep.
Just a pound or 2 in the mix.
Yep. Yeah, we did a previous on-farm study that I didn't talk about today that included tillage radish, and that performed really well. Here we went with the dwarf Essex rapeseed as our brassica, in part because it's a reliable overwintering species, and we really wanted to maximize biomass in that niche. But that's a really nice comment about, again, thinking about the complementary benefits that you get when you have a mix. Summer scavenging. And the tillage radish is a great nutrient scavenger as well. The challenge there is that it is killed by frost, and it can lead to a big pulse of nutrients actually pretty early, and that's why a mix is a benefit with tillage radish if you have some overwintering grasses that can help tie up those nutrients and then hang on to them until the spring. That's a real benefit. Any other comments, questions? These are all great input.
Oh, there's
See
one
one
more.
more.
Okay.
In you guys' work, have you seen some overwintering stuff that might be more beneficial? We've been working with Balancia clover and some things like that, trying to establish it after wheat, and it seems like it's a real battle. And I don't know if it winter kills, and I'm right up against the Ohio Michigan line,
Interesting.
and we're just struggling with it.
So the question was about reliable overwintering legumes specifically. Well, you know, the most reliable one is hairy vetch, and I know a lot of people get nervous about that one. We used vetch in another study, and I'll just give it a plug for, like, if you can handle all of that biomass and it doesn't scare you, the nitrogen inputs can just be huge. We had some farms up as high as 250 pounds of N per acre from vetch with a good stand. We've had a lot of success with the crimson clover, so that's the one that we've been sticking with for now. We also tried Balanza. It worked pretty well for us. And red clover, we've had some success and some not. Like, the issue there is a biennial. It doesn't give you quite as much nitrogen if you terminate it in that first year. But if you're able to interseed into something like wheat, red clover is a big benefit.